Hello everyone and welcome back to the genetics lecture series. Today we are gonna be learning the fundamentals of nucleic acids including DNA and RNA. The learning outcomes for this session are the following. After watching those videos and the revision, you should be able to describe the basic structure of nucleic acids, especially DNA and RNA, in terms of the sugars and bases. You should also be able to explain the difference between DNA and RNA in terms of structure, localization and function. You need to know the base pairing rules, and you need also to be able to explain the terms 5' and 3'. In addition to this, students should know the names and basic structure of the three main RNA types within their cells and understand the concept of major and minor groove within the DNA. Before we are gonna be having a closer look at the structure of nucleic acid, I would like to briefly talk about the history of DNA and RNA discovery. And the first steps in this process have been made quite early on. In 1869, a German physician called Friedrich Miescher wanted to characterize the chemical composition and properties of white cells. To do so, he isolated them from discarded surgical bandages. And he was quite lucky because he was able to publish his findings in 1871 with the title Chemical Composition of Pus Cells. He isolated what he called nuclein from the nucleus of pus cells, and he didn't know it back then, but he was basically isolating white blood cells, and in contrast to red blood cells and platelets, those cells actually do contain a nucleus and nucleic acids. He was able to demonstrate that nuclein had acidic properties, hence it became called nucleic acid. However, Miescher believed, like many others, that proteins are the molecules of heredity, but his experiments nevertheless paved the way for the discovery of DNA and RNA. Slightly later, between 1909 and 1925, Phoebus Aaron Theodore Levine identified the DNA molecules comprise a phosphate sugar base unit, and he termed this unit a nucleotide. He also proposed that DNA was made up of string of nucleotides held together through the phosphate group. Similar to Miescher, he also believed that proteins were the molecule of heredity. In our cells, there are two types of nucleic acids, DNA and RNA. And what you can see here on the right hand side is a cluster of stem cells. So each of those balls is an individual stem cell stained with the dye which is able to bind to DNA. Therefore, all of the dots appearing in cyan are nucleic acids. So what you can see here quite nicely is that the nucleus occupies quite significant proportion of the cell body. What's also quite interesting at this picture, by the way, this picture has been generated in my own research lab, are those tiny molecules you can see here. Obviously, because it's stained as well, those molecules are indeed DNA. However, they appear to be totally different to those rather round structures in here. I know we are going to be talking about chromosomes later on, but please do allow me one tiny spoiler. what you can see here is a cell which is actively dividing, and the DNA in those cells is condensed into so-called metaphase chromosomes. So this is an individual cell and this is another one, and those cells are just dividing. DNA in the cell is mainly found within the nucleus. However, there is also a small amount of DNA which can be isolated from mitochondria, and this has reasons we are going to be talking about in one of our future lectures. The second important nucleic acid within mammalian or human cells is the ribonucleic acid, short RNA. In contrast to the nucleus, RNA can be found throughout the cell, and this can be seen quite nicely on the image on the right hand side. Here the scientists stained all of the RNA molecules with the green dye, and here DNA is not stained. So as you can see here, there is a lot of RNA surrounding the nucleus, but even in the nucleus there are few green bits, indicating that RNA is present in the nucleus and in the cytoplasm. So how do those nucleic acids look like? Obviously, they are made up of nucleic acids, both RNA and DNA, and nucleic acids in chemical terms are simple condensation polymers of nucleotides. Each nucleotide is composed of the sugar. This is either a pentose sugar of a pentose sugar. We also do have a phosphate group attached to one of the carbon molecules in the sugar. We also have a nitrogenous base, and this is how a nucleotide looks like. And this is something you should keep in mind. In contrast to DNA, RNA has slightly different form of sugar within the molecule. However, what you can recognize here quite clearly, and this is the case for both DNA and RNA, that those carbon atoms are obviously at different positions, and this is quite important. As you might remember, one of the learning outcomes for this lecture is that you should be able to understand the terms 5' and 3', and those numbers are basically referring to the position of the carbon atoms within the sugar molecule. So the sugar in the RNA is the ribose. In contrast, in DNA, the sugar is deoxyribose, and the difference is basically this bit here. If you compare deoxyribose to ribose, then you will realize that we have a single H atom present in the deoxyribose, where an OH group is attached to the ribose. Again, those carbon atoms in those molecules are arranged in identical way. We are starting with this carbon atom. We are referring to this atom as 1'. Then we have the 2', 3', 4', and 5' carbon atoms, and this is the same in ribose. We have the 1', 2', 3', 4', and 5' carbon atom. So, what are the bases that are present in nucleic acids? Bases within the nucleic acids are adenine and guanine, and together those are double ring purine bases. And we also do have thymine, which is present in DNA only, cytosine, and uracil, which can be found in RNA only. And those are single ring pyrimidine bases. So please keep this difference in mind. As you can see here, nucleotides are all orientated in the same direction within nucleic acids. And the phosphate group joins the 3rd carbon of one sugar to the 5th carbon of the next in line. And this is why 3' and 5' carbons are quite important. In contrast, the bases are always attached to the 1' carbon. You can see this quite clearly here. And the order of those bases is important. It determines the genetic information of the whole DNA or RNA molecule. As you can see here, there are different bases attached to those sugars, and the sequence of those is our genetic code. But we will have a dedicated lecture on the properties of the genetic code in few weeks times. So as you can see here, we have a clear polarity in nucleic acids. We have the 5' end, and there is also the 3' end. And this is the case for both RNA and DNA molecules. The question here for you guys is this molecule an RNA or DNA molecule? In the video, I will introduce an interactive element where you will need to answer this question to proceed with the video. The obvious answer here is RNA, and the simple reason is basically the functional group attached to the 2' carbon atom within this molecule. As you can see here, this is an OH and not H, therefore this molecule must be an RNA molecule. So what are other differences between RNA and DNA? And this picture actually already indicates one of those differences you might already know. The first difference, and this is the most obvious one between DNA and RNA, is the presence of slightly different bases. While uracil can only be found in the RNA, the corresponding base in the DNA is thymine. In addition, as we have learned before, the sugar present in RNA and DNA is different. We have ribose in the RNA and there is deoxyribose in the DNA, and this is something you should please keep in mind. Another difference, and this is actually one of the most important differences, is that RNA is usually, at least in human cells, single-stranded. In contrast, DNA is usually double-stranded, at least in human cells. With this brief overview, I would like to end session 1 of the lecture dedicated to fundamentals of nucleic acid, and I'll see you in session number 2. Thank you very much for your attention.